Okay, hi, this is Ian Klein, and I'm going to be giving my presentation here on dengue virus. So dengue is a virus which falls into a genus of viruses known as the flaviviruses. And this is an emerging genus of viruses which is known to cause a significant amount of morbidity and mortality disease throughout the world. These are, as a group, these are plus single-stranded RNA enveloped viruses, and they're sometimes referred to as arboviruses. So this is just a way of saying that these are viruses which are transmitted through some sort of insect vector. The examples I've provided here are ticks and mosquitoes. These are two of the most common. And some of the examples of viruses which fall into the flavivirus genus would be West Nile virus, Zika virus, yellow fever virus, and of course the topic of this slide show is dengue virus. So just an overview of what dengue is. On the right here, we have a cartoon depicting what the virion of the virus looks like. And so we see that there is a envelope which surrounds this capsid which contains the genomic material. So just to orient you to some of these important names here, we have an E protein which studs the envelope. And then we have a matrix protein which straddles between the capsid and the envelope. And within that capsid, we have, the capsid itself is protein, and it surrounds the genome. And on the right here, you can see what the larger structure looks like. And so these are 11 kb genomes, not terribly large, but not the smallest, like something like HPV. It has a typical substitution rate of most RNA viruses, which don't have very good proofreading. And as I mentioned, there's an E protein that studs the envelope. Come back to this shortly, but this is an important target because of its immunogenicity. It's a good vaccine target. Dengue encodes structural and non-structural proteins. Some of these non-structural proteins seem to play diverse roles, and one of them would include immune system evasion and sort of modulating the immune system to be able to replicate. And an interesting phenomenon seen with dengue is this antibody-dependent enhancement, which comes from the fact that there are multiple serotypes of dengue. And because of that, the immune response is not always efficient. And so that'll be a topic towards the end of the presentation here, which I'll touch back on. So just hang on to that. So as we can see on the map here, the global distribution of dengue tends to be in the tropics. We have Central South America, Africa, and Asia and India all along the equator, which we tend to see more dengue virus infections. And so this is in part due to the fact that dengue will spread where its mosquito vector is. And so the range sort of dictates, the range of that mosquito vector will dictate where dengue can become endemic and spread to. Some of the contributing factors for why dengue has become endemic in over 100 countries would be military conflicts, so a lot of troops moving in and out of these regions of the world. Commercial air travel, as we're seeing with any virus, can exacerbate that spread. And global warming, which again, coming back to the mosquito, is an important factor. As the planet warms, we're going to see an increased range of that mosquito moving into regions of Northern Europe and North America where previously was not able to live. And with that, dengue will follow. And so touching more on the mosquito to give some more details, the mosquito which spreads dengue is the Aedes mosquito. And this falls, there's two main species. Aedes is the genus, and then the two species primarily responsible for dengue spread would be Aedes aegypti and Aedes albopictus, also known as the Asian tiger mosquito because of its really ferocious bite. And so how dengue spread or how dengue transmission looks in the wild, on the right here we see a cartoon showing this transmission cycle. Starting with the mosquito in A, it will bite an infected human who is already harboring dengue virus and is viremic. So the virus is already spread throughout their bloodstream. From there, that virus will incubate in the mosquito and for about a week. And that mosquito will then go on to bite a presumably uninfected human and transmit those immature virus particles to the human. Within a few more days, dengue fever develops from the virus proliferating within that new host, and they will become viremic. And so now that new person has virus spreading throughout their bloodstream. And a new mosquito would then come along and bite that person to restart that cycle. So looking at some of the molecular details then about dengue to remind you of what this capsid looked like. Um, it contains that RNA genome. And as with any virus, the virus has to come in with whatever its starting material is and find a way to get to mRNA to produce proteins. Now, as I've mentioned, dengue is a single-stranded RNA plus sense virus. And by convention, we call mRNA plus sense. And so dengue is already in a good orientation to begin translating to protein immediately. Not all single-stranded RNA plus sense viruses immediately translate to protein. It's not a hard and fast rule, but in this case, dengue does. It will start synthesizing proteins immediately from its genome. Alternatively, you could go to a negative stranded intermediate and then make smaller mRNAs from that. Now, at some point, the virus has to switch from making these proteins to its full-length genome. And for that, it does require this single-stranded RNA, negative sense single-stranded RNA intermediate. And so what it'll do is it'll make an opposite-stranded negative single-stranded sense RNA. And from there, then it'll go back the other way and make a full-length genome. And so what that looks like in the cartoon here that I have on the right, you come in as the virus, it drops off its plus sense RNA. From there, it can directly translate that into proteins. And then at some point, it's going to switch and make these full-length negative single-stranded RNAs. Then it will synthesize its actual genome, which is, again, the opposite way now, plus sense single-stranded RNA. It'll get packaged by those proteins it's already synthesized, and now it's an infectious particle. So how does the immune system deal with dengue? This won't be an in-depth immunology, but there is some immunology that's needed to be reviewed so that some of this makes sense. So leukocytes are cells of the immune system which will clear any foreign material or pathogens by a few different ways. Typically, we think of engulfment, cytolysis, so killing of cells, or neutralization as some of the three main strategies. The cells that can do this then are phagocytes, which are macrophages and neutrophils. On the right here, you see a neutrophil depicted with these cytoplasmic granules, and these will contain reactive oxygen species, antimicrobial peptides, antimicrobial enzymes, all of these things that it can direct at a pathogen. Another major component to this is the lymphocytes. So these are a subset of leukocytes called T cells and B cells. And these are really what, in the end, dictate the clearance of a virus. Some of the main players then are going to be, for the T cells, CD4 positive T cells and CD8 positive T cells. So these are named aptly for their protein composition, whether they have CD4 or CD8. And these are the cells which will kill virally infected host cells. Additionally, I've mentioned the B cells. B cells will secrete a soluble protein known as an antibody, which I'm sure many people have heard. And so antibodies are important then for maintaining lifelong immunity to many viruses, but also just even in the short term, they're very helpful. So more on antibodies and what they do. I've mentioned the B cell. The B cell will, during its lifetime, it will differentiate into a plasma cell. So this is the final differentiated form of a B cell. And plasma cells display surface antigen like a B cell would, but it also secretes antibodies. These are the cells which make your antibodies. Antibodies are also sometimes referred to as immunoglobulins or Igs. And they consist of a couple different components. We have heavy chains and light chains. We have variable and constant regions, also sometimes referred to as the fragment crystallizable region, or FC region, towards the bottom of it. And so to show you what this looks like, I've color-coded the heavy chain, which is the green-looking color here. And so this is the heavy chain, and it's got two of those, which are identical, and it has light chains, which are these outside chains here, and these are also identical. And the variable and the constant regions of that same molecule you can look at this in a different way and think these terminal ends here are the variable regions, and the more centered areas here, which don't change during development, are the structural or constant regions. And this very bottom portion down here is what we would consider the FC region. And so the variable region is important in that it is what recognizes the antigens. This is what provides the specificity of the antibody to its particular antigen. So combining what these two regions look like, we wind up with a molecule that can be displayed in this fashion. And so we have this combination of variable regions on the outside and then these constant regions on the inside, and you can sort of look at the difference between the heavy and the light chains. So when an antigen binds to this variable region, I've mentioned that these are identical. These regions, these variable regions on one antibody molecule produced by one plasma cell will be more or less identical. And so it's highly specific for some antigen. Therefore, even if it's slightly different, as we see here, this darker green, sort of less spiky antigen, it can't recognize that. It is not developed and it is not evolved. Well, I shouldn't say evolved. It hasn't developed to recognize that. So this is important then for controlling an infection. If you have antibodies to one virus, that doesn't necessarily mean it's going to neutralize another one. So hang on to that piece of information, because again, when we come back to antibody-dependent enhancement, that's a really important factor. So they are highly specific to one antigen. They neutralize things like viruses, also toxins. So like the diphtheria toxin or the tetanus toxin, we receive a vaccine of antibodies which are highly specific to those toxoids, and they will bind to it and neutralize it. Additionally, these can bind to the antigen and target and become good targets for phagocytes like the macrophages to then come along and see that the antigen is bound, and it will grab onto this other end here, the FC region, which is where the host will interface. The host cell, the macrophage, will interface, and it'll pull it into the cell, and it will gobble it up, basically. And so the other important thing that antibodies do is they can activate complement and some of the effector cells, like mast cells, for example. Now, these are more in-depth conversations about immunology, so I won't go into them. I'm just trying to convey the idea here that antibodies are really important in the immune system. All right, so getting to antibody-dependent enhancement. What we see here then is a model of dengue infection. And so to begin this picture, we're going to move from left to right. And at some point, this person, whoever this was infected with a dengue virus. And that is what we will call the primary infection. And so they've made an antibody after about a week or so. They've made an antibody through their plasma cells, which recognize this dengue. We'll call it dengue 1 for this serotype, because as I mentioned, there are multiple serotypes. Now, let's assume that the person a few months later is infected again by dengue. But unfortunately, this time they're infected by dengue, let's say 3, dengue serotype 3. Now, the existing antibody, which was made to dengue 1, although it is another dengue virus, there is enough difference in the sequence. There's enough sequence dissimilarity between dengue 1, dengue 2, dengue 3, 4, and 5, that antibody is not as specific to dengue 3 in this example. And so it has a hard time really neutralizing that antigen. It recognizes dengue 1 perfectly fine, and it sort of recognizes dengue 3, which therein lies the problem. It will bind to it, but not highly efficient, and so it doesn't neutralize it. From there, a macrophage, in this case they're calling it a monocyte, but let's just say it's a macrophage, will come along, and it has a receptor, an FC receptor, which will recognize now that lower portion of the antibody. And so it says, okay, I have an antibody which is bound to an antigen, and I need to take care of this. I'm going to bring it into me and degrade it in lysosomal compartments using those reactive oxygen species and hydrolytic enzymes, all those things we had mentioned before. The issue, though, is that since it wasn't perfectly neutralized, that virus then gets inside the macrophage and it is not killed. It escapes that antibody structure and it begins to proliferate. And within that macrophage, it will produce more and more progeny and it will lyse the cell. And now you have two bad things. One, the virus wasn't neutralized and it's now propagating. But maybe more importantly, this is occurring in an important leukocyte of the body. Macrophages are really important. They play a diverse role from gobbling up debris, clearing dead cells, but also they're a sentinel cell and they will detect viruses and they will sort of kick off the immune response in the absence of dendritic cells, which are another cell type. Won't go into that, but this is an important cell and it's being killed. And so I've said that there, it's taken into the macrophage and it's inefficiently destroyed. It replicates, and this is what's causing the disease. And so the evidence that we have that this is occurring comes from, well, there's a number of papers, but I've just chosen this one here. And this is what we're calling antibody-dependent enhancement. And so the conclusions from this paper here were that antibodies made during that primary infection increase the viral replication. They actually make it worse. If you have antibodies to a different serotype and you're infected on a secondary infection with a different serotype, your outcome is actually worse. And so we see that here. In the first column here, we have a response of dengue 2 with no antibody. And then in these other two, what they've done then is they've incubated macrophages. They've incubated dengue virus with a different antibody. And so then they feed this antibody dengue complex to macrophages. And what they see here is there is a dose-dependent response to this in that you have this heterologous antibody, so this non-specific antibody now are not as highly specific as it should be. And when you increase that concentration to sort of this optimal range of 4 to 40, I'm not sure what the unit is. Is it micromolar maybe? Oh, nanogram per mils. So 4 to 40 is this range where now the presence of this supposedly protective antibody actually increases the viral titer as it depicted on the y-axis here. And so you're better off in this case with no antibody. So it's an interesting thing. I don't know of other viruses that do this. That doesn't mean that there aren't, but I'm not aware of other ones. And so what does this cause? This is the disease then, dengue hemorrhagic fever. And this is characterized by an acute febrile illness. There's headache, rash, myalgia, leukopenia, which is getting back to what happens with the macrophages. Leukopenia is a loss or a reduction in leukocytes. And so as the macrophages become infected and are killed off, you wind up with less and less of those. And then thrombocytopenia. So thrombocytes are an important protein in the clotting cascade, which is a really complicated cascade. I won't go into it, but this is the reason why we call this a hemorrhagic fever. There is a depletion of thrombocytes, and so you wind up with these petechial hemorrhages, which just you bleed out from any possible orifice because you can no longer clot. That, if left untreated, will progress to what is called dengue shock syndrome. So this is, you no longer can clot, you are losing blood, and because of that, you're going to go into hypovolemic shock, and within a few minutes, you can die from that. So this is a lack of blood volume. And so just to sort of put this in perspective on the global scale, this one here focuses on India, and it says, it just shows here the number of cases and then the death rates. I'm not sure what's going on with 2019. All of these other years seem to have a large caseload and then a respective amount of deaths. I did the calculation and they're all about the same rate, so the rate isn't very different. I suspect that maybe there's a reporting issue with 2019. I find it hard to believe that we drop off from 100,000 cases to almost, nearly nothing, respectively. But the point I want to impress upon you here is that this is a major problem, and it does cause a fair amount of death throughout at least India here. And then looking at another country, this is in Malaysia, I believe. There's a lot of information, it's busy there, but you can just look here. Yeah, so again, given the proximity of Malaysia to India, I find it hard to believe that in 2018 to 2019, there's this increase, almost doubling of caseload, and then not quite doubled for the death rate. But the point is, this is a virus which causes a lot of death, more disease, and then a significant amount of death throughout the world. And in this case, this is Southeast Asia. And so, what can be done about this? There is a vaccine available right now called Dengue Vaxia. It is a live attenuated vaccine, a tetravalent. So there's 4 proteins here, which are targeted. And so it targets, I'm sorry, it's 4 serotypes that it targets, tetravalent. So it targets all 4 serotypes, or at least the 4 main serotypes. And it targets this E protein, which is on the envelope, which I had mentioned before. However, there are complications with this vaccine, and it's sort of a strange one in that it's limited use. It's limited to the use for persons only previously infected with dengue. If you have never been infected, and this is coming straight from the WHO's website, then you tend to have really bad outcomes and complications with this. There's not a lot of understanding of why that is, and it sort of goes against maybe the intuitive sense of what we've talked about. But at any rate, that's the case. So this is just some structural data showing what these E proteins look like between the different serotypes. And so the numbers aren't important. What's really maybe more important is looking at the heat maps of the polarity. And so the red's probably negative. It's usually negative charges depicted with red, and blue is positive. And we can see that they're not perfect. They do sort of follow a similar trend, but they are not 100% perfect. And so at a molecular level, an atomic level, this makes sense as to why that antibody-dependent enhancement may occur. This serotype compared to this serotype, you make an antibody to one, it sort of recognizes the other one, mostly based on charge, I'm going to venture to guess, but it doesn't do a perfect job, and it's not as efficient as it needs to be. And so future directions on where this is going, the vaccine definitely needs to be improved upon, and we would hope that they can sort of limit these off-target effects or these unwanted effects of that vaccine. Alternatively, we can develop antivirals which target cellular processes that are important to dengue's replication. So one of them, which I didn't go into, was the unfolded protein response. And so dengue primarily dengue replicates in a substructure of the cell called the endoplasmic reticulum, or the ER. And so if we can target the ER with drugs to maybe enhance some of its antiviral properties, then we can possibly get around needing a vaccine and treat with antivirals instead, or in conjunction. And so, so last would be sort of characterizing the nature of this ADE. It's not fully understood. It's a really interesting phenomenon. And with that, I think I will end here and take any questions.